Hi and Assalamualaikum everyone. I am Nura Atika Nadira binti Muhammad from Grade 7. Today I am going to present about molecular docking and computational research with my other group members. So molecular docking is a structure-based drug design method that simulates the molecular interaction and predicts the binding mode and affinity between receptors and ligands. Or in other words, it is used to simulate the optimal conformation according to the complementary and pre-organization which could predict and obtain the binding affinity and also the interaction mode between ligand and receptor. In this molecular docking, compound database is used to screen the potential pharmacophore which more convenient and also improve the efficiency and reduce the research cost. So basically, this molecular docking technology could improve the drug target predictive and capacity and understand the related molecular mechanism for drug design. There are two types model for molecular docking, which are lock and key model and also induced fit model.
In recent years, the development of computer technology, the rapid growth of target enzyme crystal structure, data and algorithm, and continuous updating of commercial small molecule database have made molecular docking a hard success in drug design. However, there are still some problems in molecular docking which should be given full attention, which is first, there are still have differences between the visual data obtained by molecular docking and the experimental data in vivo. They are and it needs to be verified in combination with other experimental methods. Second, due to the complex and diverse nature of components in marine organisms, it's still necessary to speed up the update of the database. Third, existing molecular docking software programs are complex and they need to be optimized. And last, the existing evaluation method for molecular docking technology are still immature, and the molecule with higher score may not be the best ligand.
For the first challenge is challenge in computational method for nucleic acid ligand docking. In 2008, it reported that there were already over 60 molecular docking methods successfully developed for proteins. However, docking to nucleic acid is comparatively underdeveloped. Generally, protein ligand docking program could be adapted to RNA ligand docking as RNA ligand and protein ligand macromolecular complexes follow similar physicochemical binding principles. Protein contain a well-defined, generally hydrophobic binding site, while nucleic acid offer more solvent exposed binding pocket with high charge density and polarity. These differences require modifications to existing protein ligand docking program for application to nucleic acid ligand docking. Autodoc was used with little to no modification, so its soft by doc was revised and doc 6.0 incorporated special features for docking small molecules to nucleic acids.
Okay, so for the second challenges is prediction of interactors and affinities. It is a prediction of protein interaction has dual meaning. One is predicting that two proteins or a protein and another molecule. So the other one is predicting the mode of interaction given the fact of interaction obtained by the other means, experimental or computational. So basically, docking traditionally can only read or addresses the second problem. So docking algorithms have not been specifically designed for or capable of distinguishing interacting and non-interacting proteins. So for the third challenges is co-evolutionary analysis and deep learning. It is not clear how to distinguish the intermolecular co-evolutionary information needed for docking from the intramolecular one, which is not directly relevant to the docking. Perceive a lack of sufficient amount of sequence data on protein-protein interfaces needed for the deep learning.
Hi, Assalamualaikum. I am Nurul Azmira and I will present for the challenges part. This is the fourth challenge in molecular docking, which is challenge in water solution. Protein with highly conserved structural water molecule such as HIV protease is not considerable in molecular docking. There are inclusion of water molecule, which involves scoring function and computational time to limit the decrease of freedom of a given system. Secondly, it increases the accuracy of molecular docking and gives correct evaluation water contribution to the binding site. For challenges inclusion of water, there will be current reparameterization, pole comparison involving different interacting waters, and correct discrimination of displaceable or non-displaceable water. But this challenge can be solved by using several methods, which firstly is by Monte Carlo probability, molecular dynamic of water and binding site, thirdly by hydrated docking by autodock, and lastly is free perturbation method. Therefore, it is needed to include water molecule in molecular docking to increase the accuracy of the molecular docking. It is needed to solve the inclusion of water by using the method that have been mentioned before.
Hi, Assalamualaikum. And next we proceed to the opportunity of molecular docking. And because of its low cost and perceived ease of use, the molecular docking method is widely used as a discovery tool, especially in the academic communities. Molecular docking allows the identification of potential drug targets of diseases such as protein, nucleic acid, lipids, and directing the molecular ligand target interactions at the atomic level. So, drug target is a molecule in our body that link to a specific diseases and that a drug can target to produce the desired therapeutic effect that related to the diseases or the cancer to inhibit the cancer. So this is the general workflow of molecular docking. First, we obtaining the ligands, which is the drug, and the 3D structures of the drug target, which is the protein. And then these ligands and target complexes were assigned under the protonation states and the partial charges. And then we continue with the binding site detections on the targets. It is compulsory for us to know the binding site on the targets, know the locations of the binding site of the target, so we can increase the docking efficiency. The last, the molecular docking calculations between the ligands and the drug target. So the calculation is for us to obtain the best stability of the ligand target interaction. Next opportunity of molecular docking is the developments of molecular docking for virtual screening of natural product databases. The ability of molecular docking to enable the extraordinary structural diversity of natural products to be efficiently utilized has resulted in the development of novel technology, which is the combination of molecular docking with high throughput screening HTS. So before this, the HTS screening of crude and extracts can be sometimes to be problematic due to highly polar or lipophilic components in the sample matrix. And this combination allows for the identification of bioactive molecules from natural product databases in tremendous reduction in time. That's all from me. Thank you.
Okay, in opportunity for use of molecular docking in computational tools in drug discovery is applications of combined workflows which include docking have been explored to assist different tasks of drug discovery. For example, docking has been used in tandem with ligand-based binding energy calculations and artificial intelligence or machine learning approaches to improve the prediction performance in de novo virtual screening as well as to assist target fishing, adverse drug reactions prediction and drug repurposing. Next, Different approaches can also be applied at different phases of the screening workflow to improve docking predictions. For example, molecular docking could be combined with artificial intelligence-based methods to identify suitable receptor conformations for docking. Then ligand-based approaches could be applied for rescoring the predicted docking poses. Okay, for the next opportunity, development in computing power coupled to imaging technique, artificial intelligence approaches, and protein fold prediction tools point to a golden age for the application of computational approaches to drug discovery, which can understanding the protein shape and knowing its role within the cell provides better protein structure for molecular docking, contributing to the development of specific and selective new therapeutics while also reducing the cost associated with experimentation. Okay, lastly, heuristic search algorithm, which is one of current conformational searching methods of protein ligand docking, can be applied to the global search algorithm, especially in defined region search due to limitation of calculation time. Representative of heuristic search algorithm in molecular docking are Monte Carlo, genetic algorithm, and swarm swarm doc swarm intelligence algorithm. Okay, genetic algorithm has applied in various bioinformatics due to its simplicity, high efficiency, and easy parallel processing, and has become one of the most important conformational search algorithm in protein ligand molecular docking. Swarm intelligence algorithm has higher search efficiency, stronger global optimization ability, and faster convergence speed. This method has been quickly introduced into molecular docking conformational search. In this respect, swarm-doc use the particle to full space conformation search. That's all. Thank you.